Thanks. I'm going to be speaking today about open source antibodies, which is kind of the end product of a lot of the considerations we've heard about today around recombinant antibodies and reproducibility and transparency. All of this work was funded by NIH, so taxpayer funding, and it was performed at the University of California, where we have an obligation to make the products of our research available for public use and benefit. And along those lines, I'm going to be talking about this concept of open source antibodies. And I kind of came about thinking about this in terms of analogies with open source software. And this came from an article that was published a few years ago of just the tenets of open source software and advantages that arise from the fact that, computers execute programs written in code, and cells are really biological machines that execute programs written in the sequences of the proteins that are encoded in the proteome. And open source software makes this code widely available to the user community, as does making sequences of antibodies and full validation information available. It greatly expands the opportunity for informed use and subsequent improvement of software, as it does for antibodies. So customization is a major component of this, end user customization, that's only available when the sequences of the antibodies are provided. And the idea of open source software is higher quality softwares and increased security, greater customization, and also cost effectiveness. There's estimates that open source software has an economic value of $8.8 trillion a year, and that this saves companies up to 3.5 times what they'd have to pay for licensed software. Here's a number of examples of open source software. And the objective here is that by using and enhancing the availability of open source antibodies, that we can address the issue of molecular definition of the reagent, allow for greater customization, as well as cost effectiveness and more democratization of antibody access, especially in resource limited countries. And this has been this idea came about as a collaboration between our facility at UC Davis with AdGene, the nonprofit plasmid repository, and with the Developmental Studies Hybridoma Bank at the University of Iowa. And Doug Houston's here, and we'll be talking about DSHB at this meeting. So we all know that antibodies are really crucial reagents for enabling proteomics level research. And because of this, there's been a fair number of large-scale antibody development initiatives over the years. This is in part from a review article I was involved in with Rick Kahn, who's here, and also a paper we published in the kind of Altbach special edition of New Biotechnology that was edited by Mike Tausig. And it's just a list of these kind of large-scale initiatives that have been done in the academic nonprofit sector over the years, targeting different classes of proteins using different methodologies. I'm going to be talking primarily today about the efforts that were done at our facility at UC Davis. And one of the things that distinguishes our facility is we really had a focus from the beginning, and even starting in my research lab, on developing reagents for immunohistochemistry or spatial proteomics in the brain specifically. And one of the things we do at the very first step is to include not only a protein ELISA in the primary screen, but also an ELISA on fixed transfected cells. And they're fixed and permeabilized in a manner that is meant to serve as a proxy for antibodies that are going to eventually have a high probability of working for immunohistochemistry. So right out of the gate, we can select antibodies that are up in this upper left-hand quadrant, although we try to focus on antibodies in all the different positive zones to get a diversity of antibodies that will work in a diversity of applications. So we don't only test for immunohistochemistry, but we kind of are unique in including this as such a upstream component and also such a major component in that we take typically somewhere around 100 or more candidate ELISA positive antibodies into what we consider high throughput immunohistochemistry, especially for the fact that these are done on free-floating brain sections involving paintbrushes and things that I'm not qualified to do, but that our skilled staff are able to do, again, in a fairly high throughput. And what's really amazing about the brain is, we have up to 5,000 or more different cell types. They each have extensive processes. So we really have the ability to generate a tremendously powerful rubric for assaying the patterns that the target protein ought to display in a brain section. So between the different regions of the brain, the different cell types, the extensive spatial proteomic studies that have been done on gene expression in the brain, we can really develop a powerful rubric that, when laid upon the other pillars of antibody validation, we can come up with a very robust and powerful way to analyze these, as long as it's done by a neuroanatomist who has extensive knowledge of the circuitry of the brain, the different cell types, where the processes of a cell go, because the antibody labeling pattern does not always correspond to the mRNA labeling pattern, because the mRNA is always in the cell body and the protein may be out very far away on an axon or in a presynaptic terminal. So we've taken this approach over the last 30 years, first in my research lab, like Stefan. Came out of a research lab. I ran both in parallel for many decades. And in 2005, we started this antibody facility, really serving as the NIH-funded component for the national and international neuroscience community to develop reagents that were needed. This has led to a fairly extensive catalog, not extensive in terms of a lot of the participants in this conference, but for an academic group, it led to many research publications. We have a website that chronicles all these in a searchable database. We also provide detailed information on the characteristics of the antibodies, how they've been validated, what they passed, what they failed. Detailed experimental protocols, which especially for immunohistochemistry is really where the rubber meets the road. And then we also were able to develop a low-cost distribution system where the University of California contributes by not charging any licensing fees or royalties to keep the costs down. So we've distributed a lot of antibodies through the system, saving a lot of direct costs in antibody purchase price, but we hope, because of the extensive validation, saving a lot of the staff time and more importantly, the incorrect information in the literature that could lead to wasting of valuable research funds. So one of the things we've done is partner with the Developmental Studies Hybridoma Bank as well as the MMRRC at UC Davis to make all the hybridomas available for these. So people can obtain the hybridomas, and this really is aimed at lowering the price for antibodies that are used routinely in research labs. So, a typical antibody price, as we all know, is about $400, although one of the antibodies we made a long time ago that's licensed by my previous university is selling for over $1,000 for 100 micrograms. By making these available through DSHB, we can bring the price down to about 100. But if we make the hybridomas available, you can get a CRO to make this for about $1.60 for a 100 microgram vial. And then if you're willing and able to grow your own, you can bring the price down to a very low price. And this does not include the original purchase price of the hybridomas, but kind of the recurring costs of growing these preps over and over again. One of the things where this has become incredibly important is in neuroscience. So for many decades, immunohistochemistry and spatial proteomics in the brain has been done on tissue sections, right? So this is the classical approach, cutting sections, labeling them, imaging them. But what this does is it really just basically destroys all the connections between the neurons. And it's very hard to try to reconstruct what the circuitry was in serial brain sections. And this has come about, a realization of the complexity of this come about in recent studies following this connectivity. This is a millimeter cubed volume of a mouse brain, contains 200,000 cells and 523 million connections. Okay, so we really need to try to keep the brain as intact as possible when we're doing these studies. And there's been tremendous advances in sample preparation, so now we can make brains transparent and keep them intact, allowing for imaging. The development of light sheet microscopy allows intact volumes to be sectioned optically. And image processing and other computational advances, including data processing and archiving, have really made it such that we can now begin to contemplate imaging intact human brains. And there's an NIH initiative that was just funded where one of the goals is to label 100 cleared intact human brains with 50 to 100 antibodies for each brain. And this is healthy adults. This is different developmental stages. This is diseased versus healthy. It's a very ambitious project, but it's really confronting the reality that labeling a human brain takes an incredible amount of antibody. So human brains, right, have a very large volume. The working volume is on the order of, 1.5 liters or something of antibody solution. And, back of the envelope calculations are it'll take about 40 milligrams of each antibody to label a brain. The NIH is highly desirous of any antibody preparation only being used once, not reused. So I calculated that this would take somewhere between $800 million and $1.6 billion for the price of the primary antibodies alone. So based on the previous table, right, if we can apply this $4,000 fold cost savings by making having the antibodies available for low cost production, that this will be a tremendous help to this project, and is really going to be allow this project to continue. One of the other aspects of open source antibodies is having the sequences available, and we're all aware of the value of sequenced antibodies. So we've undertaken an initiative to sequence our antibodies. We've sequenced over 8,000 hybridoma samples, and we're making all of the sequences publicly available on this website. So again, end users could take the sequences, develop their own antibody, customize it as they need. And we've also done our own work converting these into recombinant forms just by using the sequences to design G blocks, cloning them into expression vectors, expressing them, and then revalidating them basically in side-by-side comparisons with the progenitor antibody to make sure we've recapitulated the properties of the conventional antibody that was produced by the monoclonal. So we've now deposited over 1,000 recombinant antibody plasmids at Adgene, and they also provide the sequences of the plasmid. So, and more recently, they obtained funding from NIH to develop in-house capabilities of producing the antibodies and distributing them. And I think for the very first time, this means that researchers can obtain the ready-to-use antibody, which we've actually QC'd on their behalf as part of this project, each prep that they make, the plasmid that encodes it, and the entire plasmid sequence from a single source. And this combination really got me thinking about these in terms of open source antibodies and what the tenets are, and that we really need to make the antibody available in a ready-to-use form because not everybody is willing or able to produce them themselves. We've QC'd them already, and it also serves as a standard. Even if they do decide to produce them, they know they can compare their own production runs to what is available from us. There's also the renewable source of the antibodies, and whether that's the hybridoma cell, the sequenced hybridoma cell, or the plasmid, and really to ensure reproducibility and cost effectiveness. And then the antibody sequences are publicly available. And as I said, we just published a paper a few months ago, collaborators from DSHB and Adgene discussing this and encouraging people to really try to get to this sweet spot of the tenets of these open source antibodies. And then I also went back to the information that was on that previous table and just tried to figure out where do we stand as far as these mostly taxpayer-funded initiatives, which I feel should be really aiming for this open source availability for the products of these initiatives. And for a large part, there's been quite a bit of progress made on this, and a lot more is ongoing. But I hope in the future that we will be able to encourage researchers to take advantage of these existing open source antibodies, because it's really just going to enhance the transparency and reproducibility of their research, as well as the cost effectiveness, not only the initial purchase price, but the reliability and validation that comes with these highly validated antibodies. And also for those researchers who have developed antibodies to really try to move towards making them open source with a goal of, again, enabling more reproducible, transparent, and cost-effective research, and for funding agencies to continue to support and maybe increase support of the further development of open source antibody resources. So a lot of people to acknowledge. Carl Murray, I'm actually stepped down as the director of the NERMAP facility, and Carl Murray is the new director, and one of his first accomplishments was to get funding renewed for another 5 years. Great support from people at NIH, from in-house support administration at UC Davis. Our collaborators. I'd like to also thank Giovanna, who helped us with generating a specific plasmid for enhancing the variability of the antibodies we're producing, and then also the kind of gentle prodding from Andrew over the years that really, against much reluctance on my part as an old dog that wasn't quite sure he was ready to learn such a new trick, drove us into the area of recombinant antibodies and has continued to provide really valuable insights as we've moved along this path. So thank you very much, and I apologize if I went over a little.
Thank you, Jim.